Sernova is one of the many companies working on a cure for type 1 diabetes. Its unique approach implants insulin-producing cells into a bioengineered cell pouch, and in early studies has helped some people with diabetes achieve full insulin independence for years. But with challenges like immunosuppressants and scaling the technology for widespread use, there's still a long road ahead. Welcome to the show. I'm Justin. I have type 1 diabetes, and I hope for it to be cured one day. On here, I talk all things diabetes tech, news, and management. Last week, we did an entire episode on the landscape of the cure research going into a cure for type 1 diabetes. Today, we've got fellow T1D and the CEO of Sernova, Jonathan Rigby. He's joining me to share his personal journey in the diabetes and cure space, why this mission is so close to his heart, and how Sernova's decade-long pursuit of a cure is evolving. We get into the technology behind its cell pouch, the clinical trials that have already changed lives, and the exciting next steps, including advances in stem cell-derived islet cells and gene-edited cells that could eventually eliminate the need for immunosuppressants. Keep in mind that anything you hear on this show is for educational purposes only. It is not medical advice. Always consult with a physician before making changes to your healthcare. Now for the conversation. Jonathan, welcome to the show. It's so great to have you on.
Thank you for having me, Justin. It's great to see you.
Of course. Yeah, I've heard a lot about your research, your studies, your approach. In fact, I had someone on the show who was kind of giving me the landscape of cures, but getting a deeper dive in what Sernova is doing is exciting, and I can't wait for everyone to hear what's going on. But before we dive into that, I want to understand how you got involved in all this. What's your background? How did you get into research for a cure?
Oh, my word. It's been a long journey, to be honest, Justin. So I grew up in the UK, and I'm a biologist. But when I was at the ripe old age of 18 years old, I was diagnosed with type 1 diabetes. I'm highly sure that I was type 1 for many years before I was diagnosed. So I guess most of my life I've been diabetic, and so it's something very close to my heart and even closer to my pancreas. And, I've been working in the pharmaceutical biotech field for just over 30 years now. And, I started my career with big pharmaceutical companies, and I really enjoyed that. And then I figured I may be better suited to smaller companies where my efforts could yield a meaningful impact, because I get a great deal of satisfaction from working with fantastic teams that get drug products approved for serious medical diseases. So when I was in the UK, we got a couple of products approved for pulmonary arterial hypertension and for cystic fibrosis. But I had my very keen eye open and scanning the horizon in the San Francisco Bay Area, where I was fascinated by the entrepreneurial spirit, the financings that were going on, and a lot of the scientific developments. And I particularly had an interest in a company called Aradigm Corporation that were developing inhaled insulin for the treatment of type 1 diabetes in collaboration with Novo Nordisk. And so, long story short, I finagled myself a job and relocated the family from the UK to California. And that's where a lot of this began.
How did you get involved with Sernova? When did that start?
Yeah, okay. So, I moved on from Aradigm, and I co-founded my first biotech company with a friend and a colleague called Zogenix. It was a CNS company, and we financed the company, we grew the company, we got a drug product approved and marketed for migraine. We took that company public, and it went on to develop a drug for a rare disease called Dravet syndrome, which is a terrible form of epilepsy that affects young children. So that company was acquired. And then I went on to found a company called SteadyMed in the Bay Area to develop a drug product for pulmonary arterial hypertension. We took that company public, and that company was also acquired. So in between these things, there were some pretty prestigious investors in New York and the Bay Area that invested their capital into a company called Xeris Pharmaceuticals that at the time were developing the world's first liquid stable glucagon to treat severe hypoglycemia for type 1 diabetes. And they asked me to represent them on the board of Xeris, which I was happy to do. And then the company had a technical setback. And so they asked me to work on that and fix it. And with, again, surrounded by a great team, we fixed the problem. And that company went on to do a public offering as well. And the product is approved. It sits on my bedside cabinet in the room behind me, hoping that I never need to use it to get my wife to use it. You've got one as well, to get out of severe hypoglycemia. So I also on the board of a company called IM Therapeutics. They are developing a drug product to delay the onset of type 1 diabetes. I was on the board of a company called Thermalin that was developing a liquid, a thermostable insulin for countries that don't have cold supply chain mechanism systems in place. And then a few other things, but middle of last year, I decided to look for something new and exciting to do. And a colleague of mine who I sit on a board with, a cancer company board, told me about Sernova. Frankly, I hadn't heard of the company, but I started to look at the technology and this vision of developing a functional cure for type 1 diabetes, and I was sold. I looked at the data, I looked at the technology, and they asked me to join as a member of the board of directors around June, July of last year, and I happily did so. Within a short period of time, they asked me to become the executive chairman and help capitalize the company. So I said yes. And then a little bit after that, they asked me to be the CEO. And of course, I joined again because this is something very, very personal for me. And I believe in the future of the company.
Wow. Well, thank you for telling me all of that, because it's so interesting to hear people like use your background and all the work you've done in this field. It does sound personal to you, and it gives me hope every time I have someone like you on my show who lives with this disease, understands the struggle, and genuinely wants to make it better in some way. And to get to work on something that can do that is incredible. So thank you for all the work you've done so far. I'm excited about the work you're doing here. And I think what someone like you also helps give me hope with is, so many people are jaded that there will ever be a cure. People have been told for years, 5 to 10 years, there will be a cure. And at this point, people are over it. They don't think it's coming. But when I hear about people who live with this disease working on that cure, that gives me hope, because these people like you, want to see the cure because it will better your life too, right? That's a big investment. What are your thoughts on this fact that, everyone's heard 5 to 10 years there will be a cure? I mean, have you been hearing that your whole life? And do you think that we're actually at a point where maybe that's true now?
Justin, first of all, thank you for the thanks. But I also thank yourself for the great exposure of development stage technologies, drug products, etc., that you make, because that contributes significantly to the knowledge base of people in the field of diabetes, and for parents, children, caregivers who are frightened to death because of the disease that their friends, their relatives, their husbands, wives, children have. So thanks to you. And yes, my father, ever since I became diabetic, would send me newspaper articles, 3 mice in the laboratory in Liverpool Hospital are no longer diabetic, or, there's a cure around the corner. And I think back to when, literally 100 years ago, and before then, diabetes was a death sentence. You were certain to die pretty darn quickly. And I think of the scientists in Canada, in London, Ontario, and Toronto, Banting and Best, and Collip. They probably thought they could never do anything about this, but they did. The breakthrough was made, insulin was discovered, it went on to be purified. And in fact, when I was diagnosed, I was injecting myself with porcine insulin from a glass syringe that I would reuse over and over and over every single day, and terribly blunt and nasty thick needles, and no way to measure my blood sugar. So you think about where we've come since then to where we are today. And so now, I'm the benefactor of, I use an insulin pump, I use a continuous glucose meter, like I know you do. And it's a millennia of progress from where I used to be. And obviously now we're injecting recombinant human insulin, which is very, very similar to where we are now, to normal insulin. But we've got a long way to go. And so I have absolutely no doubt in my mind that one day people will say, do you remember that disease called diabetes? It's gone. And there'll be a lot of people responsible for fixing this disease, curing this disease, and helping people live their lives on a daily basis.
Yeah, I really do hope for a day when we can say that. And I hope it's during my lifetime, because I think that would just be so incredible to see. It would feel like a payoff, I think, to your work, even to my work, I think to so many people's work. It would feel really gratifying. And then I guess just to be able to have my diabetes cured would be quite nice. This episode is sponsored by Omnipod. As someone with T1D and with an incredibly active lifestyle, it's important for me not to get tied up, especially with tubes on my insulin pump. I love to go swimming, lift weights, and play kickball with friends. And with Omnipod 5, I never have to worry about tubes getting in the way. Plus, not having to disconnect helps me to stay in range during all of my favorite physical activities. That includes sexy time too. See if you're eligible for a 30-day trial by visiting omnipod.com/diabetic. On there, you'll see full safety and risk information and trial terms. That's omnipod.com/diabetic. Let's get into Sernova. Tell us a little bit about the company. Is the cure for diabetes what Sernova's always been focused on?
Yes, it has. It's mainly focused on type 1 diabetes. We are doing some work in thyroid disease and hemophilia as well. But the main focus has always been type 1 diabetes, and the concept of replacing dysfunctional cells that a normal pancreas would have. So the alpha cells, the beta cells, and the delta cells. And so the company, going back in history, developed what you can see here on the screen. We call this our cell pouch technology, and it literally is a pouch, and it's made from implantable, pliable surgical mesh. So what the physician does right now in our clinical trial is the physician will surgically implant this beneath the skin of your abdomen, but above the muscle fascia of your abdomen. Then what happens? Your blood vessels grow around this, they grow in it, and they grow through it. And we call it a biohybrid organ. So it literally turns into what we call a biohybrid organ. But you can see these horizontal channels, they are plastic rods essentially. So when this has become vascularized, the surgeon will go under your skin again. They will pull these rods out, leaving these cylindrical cylinders inside the pouch, into which he will then, or she will then, impregnate this with functional islet cells. And so that's the technology, and we've been working on this for several years now, and we are partly through the phase 1/2 clinical trial, which means you're evaluating principally safety, and also you're looking for efficacy. And the efficacy that we've been looking for is patients becoming insulin independent. And what that means, it means that you no longer have to rely on your insulin pump, on your auto-injector, on your vials and syringes. And you can go about your daily life without that, as you know, is a burdensome way of treating T1D at the moment. And in the first cohort, we had 6 patients, and all of those 6 patients achieved insulin independence for varying lengths of time. I don't want to overpromise and overstate where we are. We are still very much in the research and development stage, but we have more patients in the study now. And then next year, we'll be moving to a different kind of cell source that I'd be happy to talk to you about.
Yeah, a few of the things that you've mentioned, I want to dig deeper into. But before we do, where did the idea for a cell pouch come from? It seems so different and unique compared to a lot of the other research out there. Why was this approach important for Sernova to focus on?
Yeah, so the scientific founders of the organization, understood that if you're going to try and cure human diseases that are related to dysfunctional cells, you need to have a mechanism or a vehicle into which you can put the cells, that you're taking them from external, you're putting them inside. And one of the things that they viewed as being critical was retrievability. So if you put the cells into an organ, into a muscle, just into your skin, heaven prevent this, but should there be a problem, it's preferable to be able to remove them. So the cell pouch does a few things. It allows this containment system that can be retrieved if necessary, but it also turns into this vascularized organ. So these cells need to live. They need oxygen, they need nutrients. And if they are in something that isn't porous and in contact with your circulatory system, then there's a chance that they will die. So that's where the idea came from, and that's what we're diligently working on today.
When someone gets the pouch inserted, is it on the outside? Like, is it something you can see and feel through the skin, or is it like behind the muscles?
So you have your muscles, and then on top of that you have the skin, and there's kind of a gap between. So it goes into that gap. So it sits on top of the muscles, but underneath your skin, which allows for this prolific vascularization. And when the patients have these implanted, they don't feel anything. You can't see it, you can't feel it, and they forget about it.
Okay. I mean, I'm just, so if I was, had a 6-pack, I don't right now because I just haven't been on my routine. I've never had a 6-pack, but I've been toned. Anyway, if and when I have a 6-pack, if I had this, I wouldn't be able to see it. Like, it wouldn't be over my abs if I was that cut, or would it? I'm just curious.
No, you wouldn't be able to see it. It's incredibly thin and incredibly malleable. And it literally gets surrounded by blood vessels, so you can't see it.
I know that this approach requires immunosuppressants. Can you explain to us why they are needed in this approach currently?
Yes, well, it's an interesting question, because you and I are diabetic because of our immune system. Not a lot of people know that. So your immune system is your best friend, where it's fighting off millions and millions of bacteria and viruses and cancer cells every single day. But it can turn around and attack the body's own cells, and there are lots of diseases caused by the immune system. We call them autoimmune diseases, and type 1 diabetes is an autoimmune disease. So once upon a time, your immune system decided to destroy your pancreas, and one day it decided to destroy my pancreas. And so what we need to do, if you introduce a foreign object into a body, be that, in this case, obviously, a cell, a biological cell, your immune system has the propensity to recognize that it's not natural, it's not native to your body, and it will attack them. So, if you have a heart transplant, a kidney transplant, any kind of transplant where you're introducing a foreign cell, a foreign article into your body, we need to tamper that immune system down to stop it attacking the organ, or in our case, the cells that have been implanted. So there's a fine balance there between suppressing your immune system to a point where it's not attacking the cells, but it's still able to fight off the bacteria and the viruses that I mentioned. There is ongoing science where people are looking at coating these cells with various matrices so that they essentially become invisible to your immune system. Or there are companies looking at gene editing the cells. So once again, they become, I'm using the word invisible, but they are not recognized by the immune system. And there was a company that recently put out a little bit of good data on these gene edited cells being implanted. But that's quite a way into the far horizon.
Let's get into the clinical trials so far. So I know there was that first study. When was that conducted, and what were the qualifications for the patients who took part in it? Roughly, I'm sure there were many, but I kind of want to have a good idea of who these people were.
No problem. So the study's actually been going on for a number of years. What's been rate limiting to the progress, the cells that we are putting in the cell pouch right now, the islet cells, which are the cells that you have in your pancreas, we are obtaining those from human donors. So these are cadaveric cells, and so we can't control how many of those we can get access to. We're waiting for donors, nor can we control the quality and consistency of those cells. So the study's been going on for a number of years now, but we also signed a collaboration with a company in Germany called Evotec, and they are using induced pluripotent stem cells to manufacture human islet clusters. So these are cells that are biologically made from stem cells. They differentiate into islet cells. So that is a program that we have going on at the moment. And our hope and expectation is that next year, in 2026, we'll be in the clinic with these stem cells. So that does a number of things for us. We then have an abundant supply of cells. We know that they are all consistent in quality, and there is no limit to how many cells we can make. So in parallel to that, we've been doing our trial and looking to optimize and understand how many cells we need to put in these pouches, depending on the weight of the patient, principally. And we've been optimizing the immune suppression regime, and we're making good progress. And we are very confident that next year we'll be in the clinic with these islet-like clusters manufactured by Evotec. That's an exciting day that I'm looking forward to.
So I want to let people know also, a lot of people are going to listen to this and be like, well, how can I sign up? I want to be part of these trials. But I know that when you're taking on people for these clinical trials, it's a very specific type of patient that you want to bring on. It's not your everyday person who's got a pretty good A1C and is managing pretty well, right? Like, you need to be pretty specific. Who are these people that are part of these trials? What is their situation often like?
Yeah, so Justin, you are correct. We call it inclusion criteria. So we and the investigators in the trial specify what kind of patients are eligible to enter into the trial. And some of the things that I can disclose, these patients have to be over 18 years of age, have to have a body mass index that is reasonably in control and in range. But importantly, they need to have demonstrated a history of severe hypoglycemia and nocturnal hypoglycemia. So these are pretty sick people. However, what's fascinating, people are diligent a lot of the time. A lot of the time they're absolutely not. So we have a mix of people in the trial, some with high HbA1cs, which is a mechanism, is a test for seeing how well controlled your diabetes is over the last 3 months. So we had some people who were in the normal range, which is less than 6.5% of glycated hemoglobin. And then there were patients that were 10, 12, 13, which you will know that's a very, very high number. And what we saw in those trials, so the patients that had their A1Cs already controlled, even though it's a tremendous burden for them to control their diabetes, they have a controlled A1C. Over a period of time, when they were on the cell pouch therapy, they maintained that good glycemic control, but without having to do all of the million things that you and I have to do every day to control our blood sugar. And then the people that had high A1Cs in the 9, 10, 11 range, those came down into the normal range. So from 10 to below 6 to 6. And to put that into perspective, because it doesn't sound like very much, there are many, many studies showing that if you reduce your A1C by 1%, you reduce the incidence of cardiovascular problems, myocardial infarction, by about 14%. So bringing that A1C down is critical to the long-term control of comorbidities associated with type 1 diabetes that a lot of people frankly forget about.
These patients with this study, I know that there was one who became insulin independent for over 5 years. Where's that patient now? How are they doing? Are they still insulin independent? How long has it been? What's going on with them?
Yes, so that was actually the first patient in the trial. And this lady, whose name I can't mention, she became insulin independent at the beginning of the trial. And she was still insulin independent after 4.5 years. Unfortunately, and there were no additional surgeries, no additional transplants or implants of cells. After 4.5 years, she developed another condition, which meant unrelated to what the study that we were conducting, and she had to stop her immune suppression. So that meant that she had to come out of the trial. And so we had the opportunity then to retrieve the cell pouches, and we retrieved them, and we showed that the cell pouches had no fibrosis, the structural integrity was 100% intact. And then we went on to do histological analysis, so cell analysis of the islet cells in the pouches. And we showed an abundance of fully functional cells producing the 3 hormones that you need to control diabetes. So, people, when you talk about type 1 diabetes, they say, oh, you just need insulin. What's the problem? But the normal person on the street, their pancreas makes 3 hormones. So you have insulin that brings your blood sugar down. But in a normal pancreas, it also secretes a hormone called glucagon, which raises your blood sugar to prevent you going hypoglycemic, which is, as you know, is not a pleasant thing and is life-threatening. But then also a hormone called somatostatin that controls insulin and glucagon and keeps your blood sugar in the normal range. So what we're doing here, we're not just providing patients with replacement of their insulin, we're providing them with normal glycemic control associated with all 3 hormones that are secreted by your pancreas.
Okay. And I know that there were another, 5 or 6 patients that became insulin independent. What's the status of those patients?
So some of them are still going on. So they're still, they're still being monitored by the clinical trial site and still insulin independent. And some of them had to stop therapy for various reasons. Again, let's be realistic here. This is an early stage proof of concept clinical trial upon which we will build year on year until we get this approved.
How long are you going to monitor all of these patients?
We monitor, if they're still on the therapy, we monitor them all the time. These patients they still use continuous glucose meters, so they can see every minute of every day what their blood sugar is. In fact, our principal investigator, he has the continuous glucose graphs and charts on his phone as well. So these people are being monitored very safely, and there are obviously safety measures in place for when you're running a clinical trial, and we take those very seriously, and we monitor these people all the time.
Okay. And I know that some people failed with this treatment. What were you seeing there? Did you come up with a reason as to why some people, it wasn't taking for them?
Yeah. So in the second part of this trial, we call it cohort B. What we were trying to do was put more cells in, but also try to reach a immune suppression regime that is well tolerated. These regimes can be poorly tolerated by people, but to get it to that level where we're not causing the immune system to allow the immune suppressants, allowing your immune system to attack the cells. So, in very layman's terms, we've been turning knobs on the cooker head to try and balance all of this out. So in some of those patients, we have some of them ongoing that are insulin independent in this second cohort, and then we have others that failed. And that's what we need to know. We need to know what works, and we need to know what doesn't work, so that when we move into the next cohort, you're taking a big step forward each time you do these trials.
Are you recruiting at all right now for any trials, or are you kind of working with what you have for now?
No, we are. Yeah. If you want to learn more about this, there's contact information on our website. Feel free to send us an email.
Yeah, I'll put a link to that in the show notes. Today's episode is sponsored by T1D Exchange. You can directly make an impact on diabetes healthcare, treatments, and technology by participating in the T1D Exchange registry. It starts with a simple survey about your life with T1D, and it only takes about 15 minutes. After that, you'll have a personal portal with ongoing T1D study and survey opportunities. Plus, some of these studies even offer compensation. Signing up with the link in the show notes helps support my channel, and it allows me to continue putting out free content. You can sign up at t1dexchange.org/diabetic or click that link in today's show notes. So let's talk about kind of what's next for Sernova. You mentioned the stem cell-derived islet cells. Can you kind of explain that to us a little more deeply as to why that will be helpful? Right, like right now you're relying on donations of cells, right? So this could be an opportunity to just mass produce. Is that what this is?
It's certainly mass production, but when you obtain donor cells, so unfortunately these poor people are deceased, and they are on the organ donor list, and the pancreas and other organs are retrieved, and thank the Lord for people willing to do this. And so when the cells are harvested, for want of a better phrase, they are not always the same. Taking cells from a deceased organ and putting it into a healthy human being is miraculous. And, we've been able to achieve that, and, organ transplantation has been able to achieve that. But there are limitations on quantity, as you said, but also on quality. So deriving these islet cells from stem cells allows us to address the quantity issue, because they're made in a GMP laboratory facility. So you can make as many as you like. And they all will be the same consistency and quality. And the cells that we have so far, we've done them in preclinical studies, and they too produce insulin, glucagon, and somatostatin. And we've done some animal models where these animals are induced to be diabetic. And of course, their blood sugar flies sky high. and then they are the recipients of transplants of these cells, and then that blood sugar comes into the normal range, and we see a very flat line. Justin, if you and I could show our curves on our phones now, we don't have flat lines, but these high quality, highly functioning cells are in the normal range for the duration of the study. And even when you give the animals a high glucose diet, a high carbohydrate diet to see if their blood sugar spikes, it doesn't. It stays controlled. So it's the future. And when we get there, we can say we had a functional cure. Will it work for everybody? Who knows? I don't know.
These stem cell derived islet cells, are these one step toward the gene edited cells that you're working on that wouldn't require immunosuppressants? Kind of, are there some phases that you have to go on before you get to a cell that evades the immune system?
Yes, you're absolutely correct. So the stem cell derived islet cells will be our first entry into the marketplace, if you like, and then in parallel we will continue to work on these gene edited cells, and then the expectation is that one day the stem cells would be surpassed by the gene edited cells. Well, they're still stem cells, but they edit the genes so that the surface of the cells is not recognizable by the immune system. So that's the I'm not sure this is an appropriate phrase, but that would be the Holy Grail.
Yeah. What would you say is the big obstacle here?
The big obstacle is science. It takes a lot of time to validate the science, to scale up the manufacturing, to make sure that everything is at a very, very high level of quality, and that these cells are very, very safe. You have to prove that in many, many studies. So it is a time and a money, candidly, that it just takes a long time to do this work.
So recently, a company, Sana Biotechnology, they put out research that their approach for a cure doesn't require immunosuppressants. What did you find interesting about their approach?
That was fascinating. They implanted these cells into the forearm muscle of the patient with type 1 diabetes, and they monitored their insulin use and their glucose control over a month, over 28 days. And at the end of the 28 days, they didn't see anything of safety concern, and they did see insulin independence. So the patients wasn't not needing to give themselves exogenous insulin. So that's wonderful news, but we also have to put it into some kind of perspective. It was one patient over 28 days. So ultimately, you're going to need data on many, many patients for a long, long time. So great news, but it's very early.
Is there some form of crowdsourcing of information? Like you have Sana Biotech coming out saying they did this. You have Vertex building a factory for their derived stem cells. I'm sure you're not trading super secret stuff, but is there some form of, when you hear a company do something, oh, maybe that's a direction we kind of go into? Or is there some form of communication going on between these companies?
There is. I mean, there's definitely communication, and we communicate with people in the field. They're interested in what we are doing. We're interested in what they're doing. And, all of these clinical trials, particularly in countries like the United States, you have to get permission to do them by the US FDA, the Food and Drug Administration. So those drug applications and the protocols for doing the trials, they are public domain. So you can go on to pubmed.gov, it's called, and you can read our clinical trial design. You can read Vertex's clinical trial designs, what kind of patients they're looking at, exactly how they're conducting these studies. And then there's an obligation when you get the data, when the trial completes, that you have to disclose the good, the bad, and the indifferent. And each trial, especially early, there's good, sometimes bad, and indifferent data. So it's very, very transparent what people are doing. And then people like to publish their information and have peers review their information and present them at conferences. So it's a very transparent organization, actually.
Jonathan, thank you so much for coming on the show. I'm so glad that we got a deep dive on what Sernova is going on. It's very unique, and I'm excited to see how it evolves. I'm glad to hear that it won't affect people's six packs, so that's great. But yeah, this was super enlightening, and it's great to hear that someone like you is working on it, is very close to the project, and I'm hopeful for the future trials that you have going on.
Well, thank you, Justin, and I don't need or deserve any praise because I'm surrounded by great teams, great scientists, people that care about this, and I'm just one of the little cogs in the big mechanical clock. But thank you, and again, thank you for the exposure that you give, because it's important that people know that we are working on this. It's important for people to know the reality of doing this. It's not an easy task, but look around. There are many, many diseases that have been cured. Many, many diseases were years ago were also death sentences, but the pharma and biotech industry are fixing them in a pretty rapid succession. So all good.
What do you think about Sernova's approach? Let me know in the comments on YouTube. And what do you think of the landscape of diabetes cures? Did you watch last week's episode? If you didn't, you gotta check that out. I'll throw that in the show notes. And if there are any cures out there that you want me to speak with someone from that company, I will do my best. Sernova reached out to me, I think, and luckily we could get something down. But Vertex, I've been trying. They're not into it, but maybe there are some other companies. So let me know who you want to hear from in the comments. And if you aren't already subscribed to our blog and newsletter, I've got articles going up every week on there, as well as a newsletter coming out every other week on diabetic.beehive.com. There's a link to that in the show notes. You can click that and you can subscribe to our newsletter. That way you know what is going on in diabetic content, as well as the latest and greatest in diabetes technology news. Until next week, I'm Justin. And I'll talk to you later.